Hi everybody, welcome back to part 2 of chapter 7. So in this section we're going to look at the different types of neuroglial cells and parts of the neuron. So when you look at nervous tissue, there are two major cell types. We have the support cells called neuroglial cells, or you can call them glial cells. They are similar in structure to neurons, but they do not conduct impulses, so they're not excitable at all. And their major job is to support and protect the neurons. An important differentiation between the two is that the neuroglial cells are able to divide, so they undergo mitosis readily. They can fix themselves and they can make new cells, while neurons can't. Neurons are the cells that are able to, they're excitable, so they can take in a stimulus, and they are conductible, so they can send stimuli from one cell to another. But in general, the majority of our neurons do not divide. We have some that do, but very few. There you go. So let's look at the different types of neuroglial cells. There are a total of 6. 4 come from the CNS and 2 from the PNS. And their major job, again, I think I said, was support, protect, and they insulate the neurons. So individually, here is from the CNS, your astrocyte. An astrocyte is kind of a star-shaped cell, and it wraps around neurons, providing a barrier between the external environment and the neuron. It also surrounds capillaries of the brain and provides an extra barrier there. So this actually produces what we call a blood-brain barrier. And so its major job is to control the chemical environment of the brain. So materials just can't move from the blood into the brain. They have to make it through the astrocyte as well. So it makes it a little bit harder for stuff to get through. Astrocytes are the most abundant of all of our neurons, or neuroglial cells, not neurons, sorry. And then we have the microglia. Microglial cells are spider-like structures, so they kind of resemble spiders. These are your phagocytic cells of the nervous system, and so they monitor the environment and degrade any materials that are potentially dangerous to the nervous system. Here we have ependymal cells, again, another CNS neuroglial cell. Ependymal cells line the ventricles of the brain, and they move cerebrospinal fluid around, so they help with circulating the materials. Cerebrospinal fluid is very important. It helps to make the brain buoyant. It also helps to maintain the chemical environment of the brain, and it provides cushioning. And then we have our oligodendrocytes. Oligodendrocytes have multiple processes that come off of the cell body, and they wrap around the nerves. And what they do is they produce a myelin sheath. And myelin is a fatty tissue that insulates the neurons and helps action potentials to move faster. PNS glial cells, you have the Schwann cells, and then we have the satellite cells. So here you have Schwann cells. Schwann cells are the cells that produce the myelin fatty tissue wrapping that insulates your peripheral nervous system. Each Schwann cell produces a small wrapping. That's it, one small wrapping. And because of that, we have these little intermediate areas where there's no wrapping between the Schwann cell region or between the myelin sheath. Those regions are called the nodes of Ranvier. And they actually help in assisting the action potentials to move along neurons in the PNS. And then we have our satellite cells. The satellite cells are the cells that surround the cell body, and their main job is to protect and maintain the chemical environment of the PNS neurons. So the neurons are the actual cells that send signals to the body and allow different messages to occur or different responses to occur. A neuron itself has a cell body that contains a nucleus. The nucleus has large nucleoli in them. A nucleolus is where the ribosomes are produced. We also have Niesel bodies. Niesel bodies are a specialized form of rough endoplasmic reticulum. And then we have neurofibrils. Neurofibrils are the intermediate filament of the cytoskeleton that helps to maintain the shape of the cell. There are dendrites. Depending on the type of neuron, they can have multiple dendrites or we can have just one. But a dendrite is what sends impulses towards the cell body. And then they have an axon. And the axon is another process that conducts impulses away from the cell body towards the end of the neuron or the axon terminal. So these are your dendrites, and this is a multipolar neuron because you have multiple poles coming off the cell body. Here's the large nucleus, the nucleolus. You can see the Niesel bodies, and there's a lot of Niesel bodies, so a lot of rough ER. You can see here it's showing the neurofibrils that help in the maintaining of the shape. You can see them over here as well. And then here's the axon hillock, which is the first portion of the axon. Here you have an axon. It runs all the way to the axon terminals. So there's one, there's one, there's one. Information runs from the dendrites to the cell body and from the cell body to the axon. It always runs in the same direction in general. I mean, there's always some exceptions to the rule. Okay. And then again, here you have your Schwann cells. So this would be a PNS neuron. And these are the nodes of Ranvier in between. So at this point in the lecture, you should be able to answer multiple questions. What are the neuroglial cells? What are the four neuroglial cells of the CNS? What are their functions? What are the two neuroglial cells of the PNS? What are their functions? What are nerve cells? What are Niesel bodies? What are neurofibrils? What are dendrites and what are axons? I did not answer this one. Do neurons have centrioles? So think about the function of centrioles, and if you have to go back and look at what a centriole does, and then look at if neurons divide, and then answer this question. And if you have questions about this, definitely ask, okay? So the next part we're going to talk about is the area between the neuron and its neighboring cell. So neurons, the end process, the axon terminals, meet up with neighboring cells at a region called the synaptic cleft. They don't actually touch, so they just have that little region area, okay? And that's what, it's like a little gap where information will flow. And so the synapse is the functional junction between nerves where nerve impulses are transmitted. Myelin then is a fatty material that insulates and protects the axons of our neurons, and its function is to speed up nervous impulses. There are two types of myelin that we produce. We have Schwann cell myelin and oligodendrocyte myelin. Oligodendrocytes produce myelin in the CNS, while Schwann cells produce myelin in the PNS. Schwann cells, each cell produces its own little wrapping, and then the gaps between the cells, it produces the nodes of reindeer. The Schwann cell itself is part of the wrapping. So, and that's called the neurolema. So it's a part of the Schwann cell external to that myelin sheath, whereas oligodendrocytes can produce multiple myelin sheaths around multiple axons. They do not, they just produce the myelin. There's no neurolema with them. And so here you can see the myelin sheath, and up here there's that neurolema of a Schwann cell. So in the CNS you just have the myelin sheath. So if the neurolema, or if damage occurs, the neurolema of the Schwann cell, so it's part of that Schwann cell, would recognize the damage and would be able to fix or degrade that myelin a lot of times and then produce new myelin. Whereas in the CNS, when the myelin sheath is damaged, the cell itself that's producing the myelin is not part of that, so may not recognize that damage. So let's look at some basic terminology about neurons before we get into how the neurons work. So when we look at the CNS versus the PNS, the terminology changes. So in the CNS, when we have a cluster of cell bodies, we call that a nuclei. And so that can get confusing because we have lots of terms that are nuclei. So think about in chapter 2 when you learned about the protons, neutrons, and electrons. Protons and neutrons are found within the nucleus of the atom, which is that central region. And then you have in a cell itself, there is a nucleus that contains the DNA and most of the time nucleoli. And now we're using the word nucleus again in the CNS. So in this term, in this definition, it's a cluster of cell bodies, okay? And each cell body would have its own nucleus, so you can think of it that way, okay? But if this same collection of cell bodies is in the PNS, then it's called ganglia. When you're looking at axons that are running together, then in the CNS it's called a tract, but in the PNS it's called a nerve. So we have multiple tracts in our brain that we talk about. Optic tract is an example. And then once they move outside of the CNS, we call it an optic nerve. Okay. And then we have white and gray matter. And so these are found in both the CNS, these are found within the CNS, not, I'm sorry, not in both. White matter is a collection of the tracts. So it would be those axon covered neurons, neuron axons, okay, which is where it gets the white matter. And then the gray matter is the unmyelinated regions of mainly focusing on those cell bodies, okay? In the spinal cord and in the brain, the white matter and gray matter switch. So in the brain, the white matter is predominantly the outer region, the gray matter is inner. Whereas in a spinal cord, the white matter is the inner region and the gray matter is the outer. So we can classify neurons as sensory or motor or integration centers. So functionally, a sensory neuron is also known as an afferent neuron, and it sends signals to the CNS. And so here we have these little dendrites that would be found within the skin, that would be considered your receptors, that would pull in information up to the cell body, and then an action potential would propagate down to the CNS. So what are some types of sensory receptors? We have nerve endings that receive information about pain and temperature. We have touch receptors that receive information about different types of touch. And you can see those different free nerve endings versus Meissner corpuscles. Here we have pressure receptors, lamellar corpuscles. And we actually have two different types of proprioceptors in our joints. So the Golgi tendon, and then we have the muscle spindle. Both of these are proprioceptors to help identify the location and motion of your joints at any given time. So then we have integration neurons or interneurons. These are found within the CNS and they connect the sensory and the motor, and their job is to determine what response needs to occur, if any. Then they send a signal to the motor neurons. And the motor neurons are the ones, or efferent neurons, are the ones that carry impulses from the CNS to the muscles or glandular tissue for integration, or not for integration, sorry, for a response to occur. And so you take in some impulse, it pulls into the sensory neuron, and then is sent straight to the integration center in the CNS. Within the CNS, they determine what needs to be done, and a response moves out this efferent neuron all the way to whatever effectors, muscles or glands, are going to respond to that stimulus. So structurally, we can identify neurons based on their shape. So we have we have multipolar neurons that have multiple poles. So there's one dendrite, 2, 3, 4, 5, 6, 7, 8, all off this cell body, and then we have one axon as well. So there's a lot of poles off this cell body. So that would be a multipolar neuron. We can have bipolar neurons, so there's a cell body and there's one pole off this side and one pole off this side. So that would be bipolar neurons. Multipolar neurons are the most common neurons. So these are the most common. We find these make up the majority of the integration neurons. And they're very common in in the PNS as well. And then bipolar are not super common, but they are found in regions of the nose and eye where they take in sensory information. And then we have unipolar neurons. These are mainly found in the PNS. And their job in the PNS is to take in sensory information from regions of the skin and other things, and send it to the CNS. So at this point, you should be able to answer, what is a synaptic cleft? What is the function of myelin? What are the nodes of brain barrier, and how can we classify neurons both functionally and structurally? I'm going to stop here because this is a 20-minute video. It's perfect timing. And I'll get the third video up and running in a little bit. Bye.